Hello, everyone. Welcome to the SNRE Graduate Seminar. This is our final seminar today. It looks like my connection's a little unstable, so hopefully you can hear me okay. Today we have a presentation from Fatemah, and we are very excited. And we have, for the first time ever, a video introduction. So I will play that, and then we will get started.
Hello, everyone. Welcome to our today's seminar. My name is Mike Kowalewski, and I am a curator and professor of invertebrate paleontology in the Florida Museum of Natural History. It is my great pleasure to introduce our today's speaker, Ms. Fatemah Jamal, who is presenting her exit seminar dedicated to her PhD dissertation project. Fatemah came to UF in 2015 on a fellowship provided by Kuwait Science Foundation, and has recently successfully defended her PhD dissertation. During her tenure here, Fatemah assembled an extensive collection of echinoids and symbiotic crustaceans that live on those echinoids. In the process, she has become a proficient scientific diver and developed multiple projects focused on systematics and ecology of echinoids and associated symbiotic pea crabs. It has been a great privilege to watch Fatemah's progress as a graduate student, and I'm thrilled that she will have an opportunity to tell you more about her research. Also, I'd like to thank Fatemah's dissertation committee members, Shirley Baker, Gustav Polai, and Thomas Fraser, who have provided expert advice through the duration of Fatemah's project. Fatemah, thank you for 5 great years as our lab member, and I hope we will continue to collaborate for years to come. Today, Fatemah will tell you more about her dissertation titled Ecology and Systematics of Irregular Echinoids and Associated Pea Crabs from the Gulf of Mexico and Bahamas.
All right. Well, you are welcome to share your screen now, Fatemah, and take it away.
Thank you very much to you, Hannah, and for Michael. Let me do sharing the screen. Okay. So let's dig into this presentation and talk about the ecology and systematics of the crabs and their irregular echinoid hosts from the Gulf of Mexico and Bahamas. First, what is irregular echinoids? Or did I skip it? No. In our agenda today, I'll give a short introduction and explain how the project was conducted in general, serving all the chapters of the dissertation. And later, I'll go through each chapter separately and in more details. So what is irregular echinoids? They are way over here. They are a group of organisms belonging to the spiny-skinned a group of animals, and these include the sea stars, brittle stars, sea cucumbers, sea lilies, and finally this class Echinoidea that we were interested in, and it have the regulars and irregulars. The irregulars are the sea urchins that you see maybe on the beach or while you dive, but the irregulars are the sand dollars. And the reason why they call them irregulars is because they lost their radial symmetry that characterizes all of these groups of animals, which are the spiny skinned, and they became bilaterally symmetrical. So within this class, we have 2 orders, Clypeasteroidea, which is sand dollars, and Spatangoida, which is heart urchins. And these heart urchins look exactly like a inflated potato, or looks like a heart. That's why they call it heart urchin. And for the sand dollars, which is the common name, or I'm going to refer to them both as echinoids, they are more flatter and disc-like organisms. These organisms occurs globally and are commonly found throughout the Gulf of Mexico, the Caribbean, and the Bahamas. They are super abundant potentially. They are burrowing organisms that exist in sandy substrate that could be partially or completely covered with sand. and they can leave behind these trail marks, sometimes, not all the time. They are very important ecosystem engineers. They're playing an important role in sediment bioturbation and oxygenation and recycling the nutrients, and provide food source for various predators, and serves as homes for other organisms, such as the pneutherid crabs that we will talk about today. Now, despite this importance, we do not have a lot of rigorous studies of their distribution and ecology. Because searching IDIC-Bio records for the distribution of these petingoids and clypeasteroids, there is a gap in our knowledge, and especially here in this area in the northeastern Gulf of Mexico. And even though you see a lot of places where these were reported, these are simply reported collected materials and not detailed ecological studies. And they're mostly coming from the Hourglass exhibition throughout the 1960s and 1980s over here. And if we look at the past 20 years, we can see that the records are very limited. And this lack of data is troublesome, especially with ecosystems changing with global warming and human environmental impacts. Therefore, we want to see if things remain the same, or are they still widespread and abundant as they used to be, or things has changed. In addition to all these things, these echinoids are like little ecosystems themselves, supporting other organisms and interacting with them. They may be affected by these organisms, which may affect how they perform their ecosystem engineer services. And one of the most pervasive interactions in is within the symbiotic crab that are found in the oral side, or the mouth of the echinoid. And we can see these little crabs hitchhiking on them. These crabs are mostly found, as I said, in the oral side, which is under the animal, when you look at it from the top view. And these crabs are members of the Brachyurian family, Pneutheridae. They're commonly known as pea crabs, and within two closely genera, Disodactylus and Clypeastrophilus. The main objective of the study is to document the morphology and molecular biology of the echinoids and the pea crabs present in the Gulf of Mexico and Bahamas. And we want to understand if they are geographically specific, meaning are they only present in the Gulf of Mexico, or are they only present in the Bahamas and nowhere else? And second, we want to see if there is cryptic species. Later, we want to document the spatial distribution and abundance of the irregular echinoids only in the northern Gulf of Mexico. And lastly, we want to understand the biotic interactions between crabs and their host. We want to compare the infestation rates, the mean burden, which is the number of crabs on each host. Is the host has no crabs, or has 1 or 2 or 10 or 20 crabs? And lastly, we want to know if the crabs are species specific, meaning is one crab species living on a certain host and no other host, or it can be generalistic and occupy and infest multiple hosts. So let's see what's in the material methods. Let me explain how this was addressed. We'll look at the study sites, how sampling was performed, and how the sampling were processed for morphological, statistical, and molecular approaches. The collection took place in 3 main areas. The dots represent individual sampling sites, and these were in northeastern Gulf of Mexico and the Bahamian islands of Abaco and San Salvador. 31 sites were visited in the eastern part of Gulf of Mexico, and collectively 20 sites from the Bahamian islands. I hope this video works. Good. Here's how sample collection was made. It was made while scuba diving, and each echinoid was put quickly in a sealable plastic bag that was closed underwater. The collection then is gathered in a mesh bag and transferred to the boat and put in labeled ice coolers. The reason why we keep them in Ziploc bags or the sealable bag is we don't want the crabs to get lost or switch hosts or just abandon their host. But for each echinoid in the lab, we had to give a unique ID number and photograph them from both oral and aboral side, means the top view and the bottom, and flip them over. And then taking a subsample, hence you can see the scratch marks over here, and preserve it in alcohol for future molecular analysis, because if the animal is dead for a long time, we can't perform molecular analysis. The same thing with the symbiotic crabs that was inside each of the Ziploc bags. We put them in alcohol with giving them an ID number that corresponds to their host. And further, what we do is take some measurements for both crabs and echinoids. For the echinoids over here, we take the total length, total width, and total height or depth of the organism. For the crabs, for each crab, we take the maximum carapace width, carapace length, and we sex them. Now, for the general audience, how do you sex a crab? In adults, it's by looking at their abdomen over here. And we can see that the male has a pointed abdomen and very narrow, while the female has a very wide. And it makes sense because it needs space to carry all these eggs inside of them. We also noted if the animals were vigorous or not, if the females were vigorous or not. Not only by the shape of the abdomen, because sometimes the adults or the juveniles don't have this wide or narrow abdomen. So what we do is we actually open up this abdomen and look up for pleopods, and these are just the male genitalia, and we can make for sure that these are males and not females. If they were too small or too young to be sexed, we just count them as juveniles. Later, we need to make sure that what species they belong to, not only morphologically, after just following keys, taxonomic keys, we need to perform a molecular analysis. This is when we use the PCR, which is a technique to make many copies of specific DNA region. In our case, we used CO1, which is a mitochondrial gene. And this PCR provides a good and quick, reliable tool to delimit species. And besides analyzing exemplar specimens from both echinoids and crabs, we analyzed some specimens coming from Florida Museum of Natural History and the CO1 data available online from gene banks, so we can compare specimens with others collected from other regions. The reaction is simply made by taking tissue sample that we preserved in alcohol, and we amplify the targeted region by repeatedly going in thermal cycles through a series of temperature changes, which allows many copies of the targeted region to be produced. Later, we visualize the success of this reaction under gel electrophoresis, just like in this picture. We only send successful reactions. We don't want to send failed ones because we don't want to spend money and we know it's going to come back with nothing. So only those successful were sent to a sequencing company, and the sequences then, when we receive them, we align them and we just produce phylogenetic trees using appropriate softwares. Now let's talk about the first study about the faunas of echinoids and crabs present in the Gulf of Mexico and Bahamas. This part is going to be dealing with systematics and morphology and molecular delimitation of species. Now, the reason why I began this project with this particular study is because we need to be sure what species are there. We don't want to do any ecological or spatial biogeographical assessments or evaluation of interactions with symbiotic organisms if you don't know what's actually present there. Therefore, this is fundamental first step for any bioecological system of this type. We want to do, to make sure by both combining the morphological and molecular aspects. Now, these are the echinoids, my beauties. Based on morphological characteristics and existing literature, we found 8 morphologically different species. These include the Clypeasters rosaceus that was found in the Bahamas only, Subdepressus, both in Bahamas and Gulf of Mexico, the 2 Incobes, which is Aberrans and Mechlini, and you can differentiate between them from the notches that these are very shallow and here are very deep. There's Leodia sextiperforata with 6 holes and Melita tenuis with 5 holes. And lastly, these 2 are the heart urchins, which are the inflated ones, Myoma ventricosa, which is found only in the Bahamas, and Pelagiobrissus grandis in the Gulf of Mexico. And we only found 2 specimens in there. And Leodia, we only found 1 specimen in the Gulf of Mexico. The rest was from the Bahamas. The shapes of size frequency distribution vary across the 8 species, being Leodia being one of the smaller species in our collection, whereas the Subdepressus over here and the heart urchins being very big. Because we only collected 2 Pelagiobrissus grandis, I'm gonna exclude them from the next analysis. This is for the 7 echinoid species. These are log-log plots, which each dot represents a single specimen. These are all pooled together, although these data points coming from different sites and water depths. In the left column, we can see that within each species, there is a tight relationship between test width and test length. However, in the right column, this is not very visible. It is variable in thickness. We do not know what motivates this kind of relationship, but one follow-up study is we can explore the causes of why they are morphologically plastic in terms of height, but not in terms of length-width relationship. Now let's look at the crab species found in the system. Again, based on morphological characteristics only, we found 6 species. 3 of them came from Gulf of Mexico, and another 3 came from the Bahamas. So Rogatus, Cranioticellus, and Primitivus came from Bahamas, Stephengi, Latus, and Melitae from Gulf of Mexico. We differentiate these by looking under the microscope and looking at their carapace and the shape of it, the size of it sometimes, and if it has special notches or bumps on them. In this figure of size frequency distribution, we can see that crabs varied in size. The Primitivus over here is one of the larger species, and that's the one that occupies the larger Myoma ventricosa, the heart urchin, whereas the smaller Disodactylus melitae occupies the smaller and flatter sand dollar Melita tenuis. A note worth mentioning is these are not the size of crabs you eat at restaurants. These are crabs that are very tiny. These are less than 1 centimeter. You have to look at them under the microscope. And if you're lucky, they're 1 centimeter. Most of them were very, very small. Then again, this is a log-log plots, and each dot representing a single specimen. In the crabs, we can see a very tight relationship between carapace width and length, despite they are collected from different sites. and their morphology is less plastic than their hosts. Now coming to the results of the molecular part of the study. This compressed tree of the echinoids showing the presence of 8 species of echinoid, confirming that the morphospecies that we found are corresponding to molecularly defined species. Let me break this tree up and look at it into more details. This is the top of the tree, and the incopies were once considered to be 2 different species, but recent studies said no, we should separate them, and this is exactly what we see in the tree. Now, why do we consider them 2 different species if the divergence is very shallow over here? I believe it was 3% or 4% divergence rate. It is because they are morphologically delimited. We can say that this is this and this is that. And the molecular clustering of these specimens are entirely consistent with morphological attribute of these species. And the small genetic divergence suggests a very recent evolutionary divergence or separation between these 2 species. Now with Melita tenuis, there were no separation between the Gulf of Mexico and the Atlantic or the Panama specimens. However, in Leodia, we can see our single collected Gulf of Mexico specimen to be quite genetically distinct from other specimens from Panama or East Coast and West Coast of Florida, maybe suggesting a phylogeographic signature in terms of genetic differentiation. This should motivate further studies to look at specimens collected from this particular region. The same applies for Grandis, where our Gulf of Mexico specimens are slightly separated from population pooled from gene banks. They're most likely coming from Panama. In Myoma, there's no genetic variations, as well in Clypeaster rosaceus. However, and here's where it gets interesting, we think that there is a real genetic separation between the Clypeaster subdepressus that it is from the Bahamian population and from the Gulf of Mexico, Panama populations. Yet this genetic separation is not quite enough to consider them to be cryptic species or 2 different species. No, they're all 1 species. Moving on to the crab tree, this is the first half of the tree. Unfortunately, there is no CO1 gene bank data available for me to compare Latus and Primitivus crabs. However, if we focus on the middle over here, Disodactylus carineticellus, populations from Bahamas versus Brazil and Argentina, there is enough evidence for the presence of cryptic species, meaning these are 2 morphologically similar species, but they are 2 molecularly different species. We can differentiate between them morphologically, but molecularly, they are 2 different species. For the other part of the crab tree, where we have data from multiple regions, we can see a genetic differentiation. For example, although the Brazilian Stebangii plotted in between Gulf of Mexico specimens, we can see in the case of Melitae over here, there is a substantial differentiation between Florida's east and west coasts of the species. And the population of Rogatus from the Bahamas is slightly divergent from others from Florida Keys and Gulf of Mexico, suggesting that there is a genetic differentiation, but not enough to grant them a cryptic species level. Now, if we stand back and look overall at the screen, we can see that these species are grouped into 2 genera, creating a polyphyletic group. For example, Disodactylus melitae in the middle is between 2 Clypeaster which is genetically, or not genetically, which is, it is wrong to say that these both are sister groups. You can't say that. It is controversial, and there was molecular studies suggesting that they are the same group and our result do as well. our result reinforces the recent interpretations. So in future, they need to be synonymized, or the nomenclature needs to be revised. In summary, for chapter 1, we can see that most of our morphological species are validated molecularly, especially in the case of echinoids, where they map nearly perfectly between morphological and molecular delimitations, with one possible exception of phylogeographical differentiation in the echinoid subdepressus, Clypeaster subdepressus, which will be worth pursuing in future. And we found that there is 6 crab species and 1 possible cryptic species detected within Disodactylus cranioticellus. And finally, nomenclature needs to be revised to synonymize the genera Disodactylus and Clypeaster. Now let's move to the next chapter, which I will discuss the spatial distribution of only the irregular echinoids and only in the Gulf of Mexico, since we already validated the species present both morphologically and molecularly. So we can provide more rigorous assessment of the host distribution. These are the study sites, and the light circles indicates echinoids were found in a diving trip, whereas the darker dots indicates the presence of echinoids in a multiple sampling event. The X simply means that no collected echinoids were done. And please note this single dot and an X inside of it, which means that in the first trip, echinoids were found, and the next trip, echinoids were not found on the same site. It is pretty remarkable that none of these sites were pre-selected or planned to be visited. They were selected randomly, and in most places, echinoids were present, suggesting continuous presence of echinoids over a very large area in the northeastern Gulf of Mexico. The echinoids are known to be patchily in distribution, where it is quite a bit of, there is quite a bit of literature stating this and stating that it's monospecific assemblies, or they are dominated by a single species. And this is exactly what we saw in the Gulf of Mexico. What you see here is a distribution of samples. Only samples, at least 10 specimens, of at least 10 specimens are included in this analysis. We wanted to see how many of these samples are monospecific or close to being monospecific. In the x-axis over here is the Parker-Berger index, which is simple dominance measure, which measures percent abundance or proportion of the most abundant species. So if it's 1, it means it's monospecific population. If it's, for example, 0.7, which is 70%, it means 70% of specimens found belong to that dominant species. And what you can see here, looking at all of our sampling events, you would see a dominance of single species. Most sites has 80 to 90% dominance of the same species. And we never have a situation where we would have a dominance species represented by less than half of the population. Over here, you can see 4 of the echinoids here, 1A, B, C, and E, were widely spread throughout the region and occur in many sites. On the other hand, there are 2 echinoids, which is Leodia and the heart urchin grandis. Could be rare, but however, like in Leodia, it is a shallow burrower that looks like Melita tenuis, which is very thin and small. So it is being not present in our sampling means it is actually rare. However, in grandis, it has a different mode of life because grandis is a deep burrowing animal that maybe we just simply overlooked it during collection. This graph shows the most abundant and widely distributed taxa. These bars are medians and interquartiles ranges of 95% confidence intervals. Most of these species appear to occur on depth between 8, right here, from 8 to, let's say, 15 meters, especially to the 2 species of Incopes and Clypeaster. The smaller sand dollar, Melita tenuis, appeared to prefer slightly shallower water, but also this species overlapped with the other echinoids in terms of water depth. These may be having some bathymetric gradient differences in terms of distribution of these species, but most of them are heavily overlapping in terms of bathymetric preference. In summary, since there was a gap in our knowledge with the most echinoids in the past data being from the 1960s to 1980s, we are confident to say that what we have found is reassuring. And despite climate change and all, the good news is the same species are still present, and they are widely distributed and abundant and can be found in dense populations, and appears to have the same global ecological characteristics, like being patchily distributed and being monospecific. So they are maintaining their status of being important ecosystem engineers. Then we can say that Leota is rare. There are other 4 commonly found echinoids having large-bodied. These are subdepresses and 2 Incopes, and they dominate deeper water and more offshore sites, whereas Melita tenuis dominated the shallower waters. Next, I'll talk the biotic interactions between the irregular echinoids and the symbiotic benethorate crabs in both northeastern Gulf of Mexico and the Bahamas. Now, looking at the prevalence or the infestation rate, and what is infestation rate? It is simply the percentage of echinoids that has crabs on them. The way that you can read this graph is by these top numbers are numbers of specimens collected. For example, out of 310 Incope mechelinides specimens, we found about 60% of them were infested with crabs. We can see that infestation rates varied among species, but striking thing is infestation is happening everywhere in all of the echinoids. So it's pervasive, ranging from less than 20% here in rosaceus, but can reach up to 95.5% in Incopia aberrans. If I want to explain this, I would say that Leodia succiperforata is a very small species, which is a shallow burrower with the low infestation. And please keep in mind, because I'm going to explain this later about the host size, but in the other species having lower infestation, like rosaceus, for example, these are mostly epifaunal animals, which are living on the surface, and that might be hard substrate for the crabs to be living on. So it's providing a very not suitable conditions for these crabs, although they are infested, but not as much of infestation rate. Then this is a visualization of crab occurrence or preference. Like, what do these crabs prefer to live on these 7 echinoids? So in case of species like Lattice, we can see that they are infesting multiple species and are more general, infesting what, 1, 2, 3, 4 species, as well as Melita melitae, Disodactylus melitae. However, we can say that Rogatus and Primitivus only occupying a single host, which can be a species specific. However, it is less clear in situation where we have one host like these, because these were collected in the Bahamas and on a single site. So it may reflect our sampling limitations and not explaining the big picture. But in the case of other crabs that came from the Gulf of Mexico, like here in Disodactylus latus and Melitae, to me, I can comfortably say that these are generalistic. Although some scientists argue about that. But I have few sites in Gulf of Mexico that we can evaluate this more rigorously. Here it is. This is a figure that tells you the nicest story for different echinoids co-occurring on the same population. So if you look at the right-hand side sites, these are site numbers 16, 18, and 32. Echinoids were present, like two different echinoids were present with 2 or 3 different crabs. So the crabs have options to occupy hosts. We can see that crab Stebbengi only infested subdepresses. The crab Melitae only chose tenuous. I can't see my cursor, but however, the crab lattice remarkably can occupy both subdepresses or Melita tenuous. So it can occur on 2 different species, but in different percentages. And then we have the same exact pattern on the right side of the screen with striking consistence of crabs infesting certain hosts. But this time we have a different host, which is Mechelinae. Melitae, Disodactylus melitae crabs always infested this Mechelinae, and the crab Stebbengi always infesting Clypeaster subdepresses. And we have the kind of generalistic crab that can occupy both hosts, but with slight different percentage. As I said, some researchers argue that these crabs are generalist and others are species specific, but I think we have a strong argument over here that these are real preferences, at least in this part of the Gulf of Mexico. So here's a slide showing that there is a very strong positive relationship between host body size and the prevalence, which is infestation rate, and the burden as well. Burden is the number of crabs per echinoid. And the higher the burden, the more the echinoid size. And as far as we know from the literature, which had a maximum burden of, I believe, 19 crabs on a single echinoid specimen, we had a record. We had 29 crabs on a single Incopi Mechelinae specimen. And these crabs, of course, were juveniles and adults of different sizes and even different species, because different species can occur on a single host. And this is amazing how with host body size, we can predict in pretty high certainty whether it's going to be infested or not, and how many crabs are estimated to be present regardless of species. At the end, in summary, crabs have a host preference, and infestation rate varied among the species. And we can say that infestation frequencies and burden was positively correlated with body size of the echinoid host. In conclusion, we can't emphasize enough on the ecological importance of these irregular echinoids in the marine benthing communities, from bioturbation to various ecological services, including their importance as hosts for symbiotic animals such as pennitherid crabs. And these symbiotic pennitherid crabs are frequent infestors of echinoids found at all sample sites and present on all of our collected echinoid species, which are widespread and abundant in the study system. And because of genetic analysis and information, we now know that the fauna present at each site are accurate, and we are more confident with linking crab to host selectivity, and as well as having sites where multiple hosts are present with different crabs having the opportunity to choose a host, and few show that they are highly selective. And without the molecular data confirming that it is a monospecific species, we wouldn't know if, for example, Cranitochelys would have a cryptic species. Last but not least, thanks to all who participated or volunteered in this research. Like, without them, this project would have not existed. Thank you very much. Thank you for everybody, and I'm open for questions.
Yay. Thank you so much, Fatemah, for that awesome presentation.
Thank you.
We have plenty of time for questions. It looks like we still have about 22 people joining us via YouTube, and we have about the same number in our Zoom meeting. Please, if you're in the Zoom, feel free to unmute your mic and ask questions or type them in the chat. And if you're watching via YouTube, you can type your questions in the chat and I will read them. It looks like we have a question from Sarah. She says, great talk. I really enjoyed all the excellent natural history work you presented. Did you find any differences in host preference among the cryptic species?
No, unfortunately, I didn't study both of these cryptic species. We only studied a species from the Gulf of Mexico. The other cryptic species came from another place, which is, I'm not sure if it's Panama or Brazil or Argentina, was it? So Brazil and Argentina. So I didn't study them personally, but there is a lot of literature about Cranitochelys and their host, and it can infest multiple hosts. But most of our data is mapping on other research.
It looks like we are getting lots of emojis in the YouTube chat. Lots of
can't see, guys, but thank you.
Lots of waving and clapping.
Okay. Thank you.
Any other questions for Fatemah?
I hope you know more about echinoids or sand dollars. We commonly call them sand dollars, but echinoids may be a new term.
This is sort of a silly question. I've always heard some of our Florida, the non-flat ones called sea biscuits. Yeah, okay.
Yeah,
thanks.
It's not a silly question. They are common, they have a lot of common names. Some people call them sea cakes or something like that. But you're right. The most popular name is sand dollars and sea biscuits. Basically, biscuits are a little bit inflated, but not as the heart urchins. And that reminds me of a joke when I first came into the museum, and I believe it was Dr. Irv who asked me, what is the difference between a sand dollar and a sea biscuit? And I was trying to explain for him, but he said, no, you can't buy sea biscuits with sand dollars or something like that. It's like, okay. Okay. But yeah, it's mostly about the thickness and nothing to do with them being biscuits you can eat.
We don't get a lot of jokes in the SNRE seminar. Thank you for that.
I'm a bad. Okay.
Oh, looks like we have a question here from YouTube. It says, is there a picture you guys took of the echinoid that had 29 crabs on it? That is
I do have a picture, but I'm not sure. I believe it's, let me see. I believe it's this one, exactly. Like this specimen had 29 crabs. There's nothing special with it. We tried to do some analysis if the size, for example, or the water depth, but I never understand why there was 29. The next number on my list was, I believe, 21. Then it just goes less from that, but they're very common to have multiple up to 10. And the average, I believe it's 2, 3, or 4 crabs, depending on the species, on the crab species. But this is it's Mechlini. It's pretty remarkable, but most of them were juveniles. So I had to really look closely for these crabs because they were very, very little. You can't imagine how little they are. Even my supervisor were asking me and telling me, how on earth did you see that? Because they are very tiny. But if you look at them under the microscope, you can see that they are crabs indeed. And I'm sure I overlooked some of them, but there are a lot.
Not a question, but you did get another comment here that says, you are awesome, wonderful work.
Thank you. You are awesome. Thank you for listening. I know I talked too fast. Pardon my English.
You did great. Any last minute questions before we wrap up?
I had a question.
Go ahead.
I was wondering about, so my experience with sand dollars is like the ones that you find on the beach that are dead, and it's just kind of like the skeletal structure or whatever. And I was wondering if you only collected the live specimen, and if any crabs, maybe hang out on the whatever skeletal structure. I'm not sure exactly what to call it. But maybe there's crabs that like hanging out on those over like a live echinoid.
So we only collected live echinoids. We did collect it at some point the dead skeletons or the tests. They are scientifically called tests. And we did not find any crabs because crabs need this living organism. It's like a living island that can take them everywhere, collecting food. And maybe I didn't mention this in the presentation because I didn't study it really well. These symbiotic crabs, in general, they are called symbiotic, but if we looked at them, they are parasitic because some of them just eat the spines of the echinoids. So few depends on the echinoids as food source. But I don't argue here, maybe because I like the crabs too much, but I have to do gut content analysis just to make sure that they are parasitic. Some of them are free living, or maybe one species is free living that can be found outside of the host, but they mostly live on the living echinoids. So they are, you can call them obligate infestors.
Cool, thank you for clarifying.
You're welcome. Good question.
And you did get a couple more congratulatory notes in the
Thank
YouTube.
you.
So it looks like no more questions. So let's go ahead and wrap up for today. Thank you so much for taking the time to present to us. And this wraps up our SNRE graduate seminar for the semester. So join us next semester, everyone. Thank you so much.